So, hi, good morning, everyone. I'm Salma Rouichi, and I am PhD student at Mohammed VI Polytechnic University. I am fourth year PhD student, so maybe this is my last presentation as PhD student. So I'm very happy and honored to be here today to present you part of my PhD on exploring the genetic diversity of wild lentil species, especially for abiotic stress as drought and high temperature. But today I will present you part of this PhD on phenotyping and exploring the genetic diversity of the root system of wild lentil species. So let me answer the first question, why lentil? So lentil represent the third most important legumes crop, which means high economic is important, especially for North African and Asian countries. Also, lentil provide between 25 to 30% of protein and other nutrients, especially iron. So that's why we call it nutrient-rich crops. So as you know, that lentil, as the other legumes crops, contribute to the nitrogen cycle by fixing atmospheric nitrogen in the soil, which allows them to grow in almost all types of soil. So they are very important in crop diversification and key components of cereal-based cropping system. So to give you a little bit context and background for this study, I want to talk a little bit about the challenge for lentil production, but I will not talk about climate change. I think that all of you know the impact of climate change or on agriculture production. But I would like to talk about a challenge which is very specific to lentil, which is the narrow genetic base. So here I present numbers. We have less than 50 varieties in 14 countries in the last 10 years. That means that we don't have much work, development, and improvements of new varieties for lentil. Also, the domestication process contributes to this loss. Almost 40% of the genetic diversity of lentil has been lost to domestication alone. So here to present you the last genus for lentil. So we have 6 species. We have 4 gene pools. The first one with 2 species, I will say name briefly. We have Orientalis tomentosus for the first one, the second gene pool with 2 genotypes, the third one with Lens ervides, and the last one Lens nigricans. But these species are not fully explored for different traits. So we have untapped genetic diversity of this wild lentil species. So my PhD started with this modeling study. We used large data set for wild lentil species to do species distribution modeling. We used machine learning algorithm to assess and evaluate the impact of climate change on the distribution of this wild lentil species. And the idea was to identify species or genotypes that showed good resilience under different climate change scenarios. And why? Because we believe that this species able to cope with different climate change scenarios for sure potentially have a trait that make them resistant in the past in their centers of origin and continue to make them resistant under different climate change scenarios projected for 2050 and 2070 based on this modeling study. After that, we selected from this modeling study, we selected 61 wild lentil genotypes coming from different regions. As you can see here on the map, the distribution of wild lentil species is very large, so we tried to cover a large part of this distribution. So we screened this wild lentil species for physiological trait called water-saving trait by limiting transpiration rate under high vapor pressure deficit, or what we call high temperature, more simply. So we found 2 important points. The first one, we found that we have large genetic diversity of wild lentil species for this trait. Secondly, we found that wild lentil species have the capacity to control transpiration by closing stomata early in the stress period. And that allows them to conserve water in the soil to sustain development and growth. So we said maybe this is one of the survival strategies adopted by wild species in their centers of origin or in the native areas. So after these 2 studies, we said that we wanted to dive deeper into understanding of this wild lentil species. So we said that we have to look into the variation of the root system of this wild lentil species. So interestingly, we didn't find much work. I think that we have 3 or 4 studies that studied the genetic diversity of this wild lentil species, but First of all, they didn't really use large data set for wild species, and also they didn't really use high phenotyping techniques. So we said that we have a huge research gap here. We have untapped genetic diversity when it's come to the root system. So now coming to the root phenotyping, and I think that as many of you who already work on root phenotyping knows that this area comes with several challenges, especially when it comes to the limited visibility. It's really hard to visualize, quantify the root system in the soil. And also the technique we used before are often destructive techniques, so we have to remove roots from the soil to do our analysis and quantify the root system. And also time-consuming, and we require more efforts. But recently, with the advancements of root phenotyping, this, as shown here in the figure, we have more and more efficient techniques to phenotype the root system. So now I would like to introduce you the root phenotyping platform that we used. So it's called the Rhizoscope. It's advanced phenotyping platform located at CIRAD in Montpellier. So this platform, it was designed to phenotype annual crops as wheat, barley. I think they already used rice, lentil. So it's fully controlled. We can control all the environmental conditions, our temperature, lighting, humidity also. So it consists of 4 containers. Each container can hold up to 50 Rhizobox structure, and each Rhizobox, it's a structure made of 2 plates, each one measured to 50 by 20 by 2 centimeters, and filled by plastic beads. This plastic beads was to mimic the soil resistance, and also, And also we have the most important thing about this structure, that we have pin structure. I don't know if you can see it, but you will see it in the next, yes, you will see it here in the next slide. This pin structure is very important because it helps to conserve the stability of the root system after we remove the beads, and also conserve the spatial distribution of the root system and the rhizobox. So the second component is what we call rhizomaton. It's a dark chamber where we can take pictures, images for the root system. So it's conserved with, it allows that to have the image with the same light intensity and also with the same distance between the rhizobox and the cameras. So after that, we analyzed the image. We developed a Python algorithm with CIRAD team. So here we have the basic traits for root, for example, maximum root depth, root angle, total root length, etc. And also we developed traits that we said that these traits are very important for white species. For example, we have the collar-ramification. So we observed that here, for example, we observed that this length between the collar of the plant and the first ramification was very important in wild species compared to the cultivated species. It goes up to 8 centimeters for wild species to have the first ramification. Also, we studied the root density in more detail, so to understand the spatial distribution of the root system in the rhizobox. So we have the mean density, the average of the root density. Also, we calculate the depth corresponding to the maximum of root density and trait corresponding to the spreading or the spatial distribution of the root system in the rhizobox. So here for the root density, I will explain very briefly how we calculate the root density. We stack the image to have the same, using the Python algorithm, to have the same X and Y axis, and we did all the root segmentation to remove the background and also to transform this image to the binary image. So we have 0 for non-root areas and 1 for the root area. And we average the root density across the x-axis. So we have the root density over the x-axis for 5 millimeter depth for the root density. So here we have cultivated genotype, and here we have the wild genotype. So here you can see the pattern of the root density across the depths. This is the cultivated genotypes, and this is the wild genotype. So here we can extract all the information on the maximum root depth and also the pattern of the distribution of the root system in the Rhizobox. So now coming to the results, we started our analysis by doing PCI analysis with all the data, and the idea was to see how these genotype clustered, and also to see which variables or root parameters are responsible for this clustering. So here we have 3 clusters, but basically we have 2. We have one cluster for cultivated genotypes and 2 subclusters of wild species. So here we found that cultivated genotypes had high values for root angle, root mass, root volume, and also density, mean density. That means that cultivated genotype had larger and deeper root system compared to wild species. But more important thing that we found that wild species has higher values for root-shoot ratio. That means that we have the cultivated genotype that have a deeper root system, and in more other words, that cultivated genotype puts resources on both shoot and the root development or biomass, and we have the wild species that develop more the root system over the shoot system, and having higher values for. And I have to say that our experiment was not conducted under water-limited conditions, so it was like screening. And still, wild species prioritize the root system developments over the shoots. And as Professor said, that the root-shoot ratio is functional balance. It follows the functional balance, which is the basic fundamental of crop growth models, that when we have limited soil resources, plants tend to develop more root system to access those resources, even if it means slower shoot development. Also, we found that root angle was wider in cultivated species compared to the wild species that have very narrow root angle. And also we observed the pattern that further you move from the first gene pool to the last one, more the root angle became narrower. And these patterns was also observed in wild barley. And we said that maybe this is one of also the strategy adopted by wild species to capture in their centers of origin. So here another visualization of the PCI analysis. So here we found that one cultivated genotype clustered with the wild species. So we look at it to the pedigree of these cultivated genotypes, and we found that this genotype had genetic background of wild species. And more interesting thing that this wild species coming from the same region as the wild species that we use. It's mainly from the fertile crescent regions. Here we have the root density analysis. So here we have the root density over the Rhizobox depth, and here we have the normalized root density by dividing all the root density by the maximum, so we can compare between species. So here we can see that cultivated genotype exhibits a different pattern from the wild species. We have higher root density compared to the all wild species, and here we can see that the standard deviation of cultivated species, the density for cultivated species is very it's not very important compared to the wild species. That means that cultivated genotype have uniform root density compared to the wild species. So we can see here that the standard deviation is very important in wild species. That means that wild species have more genetic variation for when it comes to the root density. So after that, we said that we have to look into the wild species. So we tried to study only the 42 wild lentil genotypes, and we choose 5 traits: total root length, maximum root depth, root angle, because those variables were chosen based on high and significant variance confirmed by ANOVA. So we can see that we have 3 clusters, one characterized by high values for root angle, maximum root depth, and also we have the second cluster, which is the opposite of the first one. That means we have 2 strategies of root system, and we have genotype here between the 2 clusters. So here we can see this is the most important thing. I think that we have here the mixture of the clustering. For example, like Orientalis, we can find it in the first and the second and the third cluster. The same thing for Tomentosus. And also the most important thing that Tomentosus and Orientalis are from the same gene pool, but they exhibit different roots strategy or root system strategy. And that means that genetic similarity or genetic distance only cannot explain the root density. So we have to look into their centers of origin and the environmental factors and their centers of origin over the time. So as a conclusion first, so wild lentil species have very large genetic variation. And as I said in my first slide, that it is like a walk on the wild side. My thesis is like a walk on the wild side because we focused on wild species, but we tried to look into different topics, started by modeling study, physiological study, and also the root system. So I can say that the wild species have very large genetic variation when it comes to the root system or also the physiological trait as water-saving traits. So we have to explore this genetic diversity. Secondly, the genetic distance or the gene pool or the genetic similarity alone cannot explain the genetic variation or the variation we observed for root systems. So we have to take into consideration their environmental factors or also the interaction between the genetic basis and the environmental factors in their centers of origin. And also we have to take into consideration the local adaptation to the specific ecological pressure in their centers of origin. And as perspective, we have to not only explore the genetic diversity, but we use this genetic diversity in our breeding programs to integrate these desirable traits to cultivated species. So at the end, I want to thank the organizing committee for this opportunity. I would like also to thank CIRAD for giving me the opportunity and also the infrastructure to do this experiment, especially Roman Fernandez, who helped me a lot in the image analysis. And also I would like to thank the Phenotyping team in Morocco and also thank my supervisor for their help and support. And thank you all for listening. Thank you.
Okay, thanks so much. We're running a little bit late, but I said you got to do my dirty questions in Salma. So you want to start us off right here in front. Okay.
Your system looks wonderful. Could you tell a little bit more about the beads that you use? Are they how big are they, and how do you water the system? And can you also use the system for drought experiments, for example?
So our experiment was hydroponic experiment. So it's the Rhizoscope was fed permanently with the nutrient solution, and at the end of the experiment, we removed the plastic beads. But our experiment was not conducted in water stress conditions. That's why I cannot say conclusion about this wild lentil species can maybe have good resistance under drought conditions. It's more like screening and phenotyping of the root system of wild species.
I understand, but could you, in your next postdoc, could you use it also for drought experiments, or is that not feasible in that system?
Sorry, I didn't get your question.
Can you use this system also for drought, or is it because it's a hydroponic system?
No, yes, we can use it for drought resistance. Yeah.
How thick are the beads?
I think 5 millimeter. Yeah. Thank you.